Okay, so I think now in the spirit of our program today, we will switch to English and we will start the presentation of our study plan. So the title of this master's degree is Biology for Translational Research and Precision Medicine. And of course, from the title, we understand that what we want to talk about is made of two parts, one being translational research and one being precision medicine, which are strictly interrelated, but in fact, they are two different things. And I would like to give you an example about what one of the two is and what of the other is to see what we aim to do in our actually master's degree program. So our goal in the end is to train a new generation of biologists that can play an active role in research in both this field, so translational research and precision medicine. But I would like to give you two examples to understand what has been the progress in this field in the last, I would say in the last 20 years. And the first one is about coronavirus. We all know that we got out of this pandemic. We are actually going out of this terrible pandemic period. And this has been possible, of course, because of vaccines, as we all know. But to produce vaccines that are effective for the treatment of this or for the prevention of this disease, we have to start from the knowledge of what coronavirus is. And to understand what a coronavirus is, we need basic research because we need, for example, to see how the virus is made, what are the proteins, you know, in the viral particle that are recognized by a receptor. And this is just basic research, essentially. We need to know how these proteins will mutate, how fast, and how the interaction occurs with the receptor. And this is basic research. And this research becomes translational when all these aspects are put together, and then there is drug discovery, and then there is the development of the vaccine, there are epidemiologic studies, and so forth. And then this basic research becomes translational. So translational research is essentially all that aims to transform results of basic research into something, some tool that can be actually very effective for the prevention, for the diagnosis, and for finally the therapeutic aspects that we want to aim at. And this is, I think, a first example that we all understand pretty well about the power of translational research. And about precision medicine, well, when you think about precision medicine, you think about something that is person-specific or patient-specific, right? And so each of us has a certain genome, has his or her own DNA. So, and we know that in the last years, it has become possible to edit this genome using CRISPR-Cas technology. We all heard about that. But what people sometimes do not recall is that the actual discoverer or CRISPR was a professor in microbiology, a Spanish professor, Francis Mojica, who was working at the University of Alicante, who at some point was studying how bacteria in archaea would adapt to very salty environments. And he found that these microorganisms would have in their DNA some repeats. And that was pretty strange. And then he did very basic research. Honestly, back then, only few people cared about these repeats. And studying archaea and bacteria was not so cool. But then this was so important because that was proven to be actually the sort of innate immune systems for bacteria and microorganisms. And then many other researchers became very much interested in this story. At the point that, as we know, Jennifer Doudna and Emmanuel Charpentier got awarded the Nobel Prize in 2020 about this technology that was then discovered, this CRISPR-Cas technology, by which we can sort of specifically cut our DNA in specific parts and correct it. And this is, again, a story that tells us how important basic science is also for medicine. And I think these two stories describe exactly what we would like to do with this new program. So really give you all the knowledge and information and the attitude so that you can do your contributions finally in this beautiful topics. So how we want to reach this goal and why you should, if you're interested in these topics, enroll in this program here at the University of Verona. Actually, there are some other master's degree that focus on some aspects of translational research or precision medicine. But actually, this is the only program that is providing you some details about these two themes all together and tries to interconnect these two realities. And this is really unique in Italy. And I would say also, if one has a look at other European and extra-European offers, this is a pretty unique program. And how we want to aim to provide you with this training, with this knowledge, we want to use our local expertise. So it is important to bear in mind that teachers in this master's degree are scientists. All of us are researchers. We do research in our labs, and we know the topics that we want to teach, or not only theoretically, but we know also the tricks and the issues that these have when we want to do research on these fields. And we will then use this expertise and inspire our lectures, the content of our teaching on our real life research type of activity. And we hope that this will be appreciated by the students, because then they will immediately enter real biomedical problems. And this is also important because we want to use, as Professor Schena also mentioned, some new ways of doing teaching, not just face-to-face lecture, what I'm doing now, I'm talking to you, but we want to have in this master's degree, and you will have in this master's degree, on one hand, a strong lab activity, strong experimental activity. And then the professors in each that teach specific classes will tell you something about their course and how the lab is important in their course. So there's a lot of lab, and this lab is really inspired by real biomedical research issues. So we start from that problem and we try to give you the tools to contribute to this, to investigate this problem. And also we want to facilitate these activities in small groups. So you will work in small groups of students, and you will then learn how to basically play in teams, because any research activity cannot be alone nowadays. It's always a team that is investigating some issue. And so if you start working in teams from the very beginning, this will be extremely useful for your career. And to do that, so to do this teamwork and this innovative sort of teaching formula, we want to use our centers of, or let's say our excellent facilities at the University of Verona, which are mentioned here. Centro Piattaforme Tecnologiche will provide all the technology. Technology is extremely important to improve knowledge. So CPT will provide the platforms, the technological platforms that allows you to use cutting-edge methodologies that are state-of-the-art to progress knowledge in biomedicine. Then there will be l'URM, Laboratorio Universitario di Ricerca Medica, which is also extremely important in our university for medical research and for translational research. So when we want to bring something from the bench to the bed, in a way, we first have to analyze the potential, the translational potential of these activities. And this is what people at the LURM do, and LURM is extremely enthusiastic in collaborating with our project. Then, of course, research and medical research has implications about ethical issues, obviously. And we have a very important center, CIRSA, Centro Interdipartimentale di Servizio alla Ricerca Sperimentale, it's a long word, CIRSA, that has an extremely high knowledge and expertise in bioethical issues and in aspects that are extremely necessary when you want to, for example, write a scientific grant to get funding for some research, and how you have to deal with some of these issues. And so they will also collaborate with us for these aspects, including alternative research models and so on. And finally, we have ARCNet, which is a very important center, a multidisciplinary center for extremely relevant research in precision oncology, and they will offer us also their expertise. So I hope you understand that this is very unique offer, because there will be classical labs, but there will be centers where you can get practice, where people do research for real. And this is a rather unique opportunity, I think. So getting into our educational proposal, it was already mentioned that we want the students to be actively engaged in learning, not just passive, like they listen and they study after 3 months and then they don't remember anything of what has been told in the class. This is not what we want to do here. We want students to be very active, actively engaged. Of course, they work in teams. The day after, maybe 2 days after, we will ask if they have studied what we have explained before, because knowledge is built in the class, not only when you study on a book at home. So we will actually experiment alternative, or what we say modern, pedagogically proven, very effective ways of teaching. For example, flipped classroom, team-based learning, challenge-based learning. So all these methods will be applied to some parts, not to all, but to some parts of our teaching programs. And this will really be effective in making you students very engaged and very independent. It will help you to create your own ideas and your own way of thinking in terms of biomedical research. Of course, this is tagged as an international study program, and therefore all our teaching activities will be in English. So English will be the official language for this reason. And since our program is quite well structured, it requires some infrastructure and some logistics and to support the activities, then we have a limited access to 40 students. This means that, as we will see, you have to apply for enrollment, and we can have up to 40 students at least for the first year of our master's degree. Then let's talk about more specific content. What do we want to teach in this new study program? Actually, there is, a master's degree follows a bachelor degree. So you may come from different backgrounds, different bachelor degrees, laurea triennali. We will make sure that you have a basic background. So this will be one of the requirements when you apply for enrollment in the study program. And then, of course, our lectures will start. And the first year, it is made of 2 years, clearly, this master degree. The first year it has a specific path. We decided that you won't have many choices in terms of different classes, because we created a path that we believe is essential for then make you independent in the choice of the classes in the second year. So the first year will be common, and it will be necessary for building up this cross-cutting basics. And then, as I said, in the second year, there will be a more specific path of specialization. There will be elective courses that will allow you students to specialize in 3 different areas of your interest that you will have heard about before. These 3 areas are molecular neuroscience, this is one possible choice, precision oncology, or laboratory medicine. So these are sort of 3 paths that you can build, and you can, of course, prepare your final project about. And this will depend on you, what is better or best for your own interests. And therefore, there will be classes, there will be courses that you can choose that will define one of these three paths. Okay. How to enroll in this study program. So I mentioned that there are some sort of eligibility requirements. So of course you have to hold a bachelor degree, because this is a master degree. So you're supposed to have first some other piece of paper that you have gained before. And this must be in some specific area. It couldn't be in scienze biologiche, biological science, biotechnology. This is already eligible. So if you have this, it's okay in terms of minimal requirements. Or you can have also a degree that has been obtained abroad, but somehow is equivalent to these programs. You can have also another type of bachelor, laurea in tecniche di laboratorio biomedico, for example, this class SNT3 in the Italian system. That will also allow you to apply for the enrollment. Or if you do not have these requirements, you can have another bachelor or even master degree, or even laurea ciclo unico, for example. But you have to have at least 60 of these credits, GFU, which are essentially identical to the European ECTS credits in some specific topics. I won't go into details now, because you then will find all the information in the website. In the bando di ammissione, you will have all the information. So it's a bit actually boring to go all to this 20 CFU di questo, 15 dell'altro. There are possibilities if you don't fulfill these requirements to apply anyway. And this will be explained in much better detail when you look at this bando di ammissione. Okay. And also, there is a selection that is based on your graduation mark. So there is a minimum in the Italian system that will be a minimal mark of 90 su 110 for the bachelor's degree. There will be some equivalent when one applies from, of course, foreign bachelor degree. We will make some comparison, but this is a minimal requirement to be eligible for our program. And there is also a sort of evaluation of your CV and a very simple interview. It's nothing dramatic. It's just a little interview that when we want to test what, for example, you did in your bachelor program, or how your English is, and something like that. So it will be a very simple interview. So no panic. It's nothing very difficult. So all this together will create the requirements for you to be enrolled in our study program. Of course, if there are questions, then we can specify better these requirements. But I would also recommend that you have a look at our website. It's already online. So here you see the web address, and there is both the Italian and the English version. So you can get all the information that you require from the website. It's already quite complete, I would say. So I really invite you to have a look at it. Then before entering the actual study program, of course, you want to know what these classes are. But before that, I would like to give you just a few hints about what you can do after you graduate in biology for translational research and precision medicine. So what are the main employment outlets or job profiles that would match your experience and your expertise? So you will become senior biologists with an expertise in translational research and precision medicine. In practice, this means that you will have a specific knowledge in basic and translational research, and that you can use in biochemical, but also in clinical and health fields. So in fact, you can work, as probably it's later, in public or private research institutes, but also hospitals or structures for health system, for example, or in a more basic research lab, if you prefer. You will have also competence in personalized medicine, topics like advanced diagnostics, development of prevention, diagnosis, and therapy tools, all that you will have learned from your studies. But you will have also some background into actually some very important skills that are more and more requested by also by the companies that work in this field. For example, how to plan an experiment, how to plan scientific experiment, and how to manage scientific research program. These are skills that people who do research must learn. And normally they learn it quite late because they have to. At some point, you have to write the grant, you have to know what to write, what not to write, and so on. But we think it's good that you get some, at least initial knowledge already from the undergraduate school. And that's why you will have also some of these skills if you enroll in our program. And then, of course, you will have some innovative solution for the knowledge and technology transfer in the area of precision medicine and translational research. Indeed, you will have the possibility to work in public and private university, research institutes, hospital companies, in different topics: biological, genetic, quality control analysis, pharmaceutical and biotechnology industries. But also, since this is very important, since you will have developed, I think, rather unique communication and teamwork skills compared to other degrees, then you can use this knowledge to, if you're interested, to find employment in the field of scientific communication, for example, or dissemination of scientific information, scientific publishing, and so forth. And this is, I think, also another important opportunity that you will have if you successfully complete our program. And of course, you can also continue your education by applying to a PhD program, and also this postgraduate schools. We have many here in Verona. There are others in other universities. So this is, of course, the idea that if you really like to continue to do research at the academic level, you can continue with this path. Okay, then I think with the general introduction, this is all I wanted to say. And then I would like to go a bit more into details about our study plan, so that if you have questions, you can ask also from home. I don't see now the people, but I know there are some students connected. I know they are there, even though I don't see them at the moment. So of course, this is the same. If you want to ask questions also in Italian, it's fine. In the end, I think it's important that you have understood really what we want to teach you. And that is why finally I'm inviting my colleagues here, not only me talking. That is good. So we will present now the first year, and then we will switch to the second year of our master's program. And as I already mentioned, we have the first year, there is a specific path. It's already decided. But as you will probably notice, it's quite well, hopefully, built, so that you will really, from the very first year, build up a good knowledge that will be necessary for the second. So, and I just wanted you to notice from this table that many, not all, but many, almost every class in the first year has some credits, which has laboratory activity. So there is always, as I said from the beginning, some more, I would say, theoretical, it's not really appropriate, but theoretical knowledge, but there is also some practical part that will complement and integrate this more theoretical knowledge. So I would then start. I think we have to switch to the other targette. I don't know the English for that. And then I would like to start from the first course, which is molecular and cellular basis of disease. I think the speaker for this, speakers for this course are Professor Elena Zenaro and Professor Giuseppe Busetto, and they will give us some information about their class.
Thank you very much for being here, first of all. The first semester, we will have this kind of big course that it will be shared by different professors. So the first, let's say, module, it's the cytology and histology that will be given by Professor Bertini. And this is kind of the basis, is giving like the general knowledge of morphological and functional study of the human body. This course can be given in many different ways. So traditionally, it's carried out just giving a systematic view of all the components of the cells and the tissue. However, in this course, we decided to limit the selection of the cell types that will be presented, and we kind of integrate all the topics together using flipped classroom, team-based learning techniques, and then also computer tools would be used with virtual microscopy preparation. And then there will be like Q&A sections to just interact a little bit more with the students and to acquire the knowledge in the classroom much better. And then there will be the second module that will be given by Professor Busetto. And yeah.
Thank you, Elena. Thank you. All right. Hello. I first want to thank Daniele for the very beginning of his introduction, because as we see science, there is no basic science that does not have a potential translational interest. Any study, anything that you can study has this potential for the future. And this is very important to stress, and that our students will learn this from the very beginning. I will teach biophysics, which essentially is the function of the membrane. And at its core, it is based on the function of ion channels. And studying ion channels from the very basic point of view has a potential for the medicine, because over the years, many diseases have been shown to be based on problems in ion channels. And now we know this as channelopathies. One for all is cystic fibrosis, which is very common and famous. So we need to know, the students need to know what a channel is, how many different types of channels, ion channels, are embedded in biological membranes, and what they do. How they are operated by modulators or by the potential of a cell, in order to see the membrane as a very important part of the cell, not only because it has receptors for antigens and many things, but also because it has channels. That's all, basically.
Okay, thank you very much. Oh, sorry. It was not over. Sorry.
It was not over.
I didn't read that.
Yeah.
So.
We had we added a third module that will be shared with Professor DeSantis, and is the cell pathology, inflammation, and immunology module, which is quite huge. And this will cover like the fundamental of the state of health and diseases. So we kind of give a broad view of all the knowledge that are necessary to understand what is happening during cellular pathology or molecular pathology. And we will have a special focus on elements of oncology and, let's say, neurodegenerative diseases. The module will provide like general information, what is an inflammatory process, including the anatomy and the physiology of the immune response. Then we will give knowledge about leukocyte recruitment, the role of the microbiota in regulating the immune system, and then all the fundamental principle of the immune response in the pathology. And then Professor DeSantis, that is working more like let's say, looking at the immunology response in the oncology with an oncological point of view. He will teach more about the biological mechanisms that are regulating the immune response in this field. and with specific techniques and knowledge apply to develop precision medicine to treat cancer. And then I will integrate this with the topic, let's say, of the neurodegenerative condition, the autoimmunity, and the inflammation in general. So at the end of the course, the point is to give like the possibility to the students to understand what is an immune response, and also to understand all the techniques that are used in order to study the immune response taking place in the different inflammatory context. At the end, we wish also that the students will be able to interpret like the manuscript that they read. They will be able to write effectively a manuscript, and then they can effectively also communicate with the scientific community. So we hope that we will be able to cover all this during our course. Thank you.
Thank you very much.
Yeah, I'm done.
Okay, thank you very much, Giuseppe, this explanation. Then we will continue with this biological macromolecules, interactions, and networks. So there are two teachers involved, one being me and one being Professor Marino. So I will give you some information about the theoretical part of this class. Maybe I have to do this. This camera is terrible. Or maybe we do this, so we have both. So as the title suggests, this is a new kind of course that aims at providing a very specific view about molecules. Normally, we think about molecules as individual entities. So we study one protein or one DNA and so on. But of course, biomolecules work together. This is for sure. So the idea here is that we start from a very detailed characterization of molecules, talking about their structure, their biochemical structure, and their function. And then we build up complexity using a bottom-up strategy. So we will first see how proteins interact with other proteins, with DNA, with RNA, with lipids, with membranes. And then we will start from individual molecules, interactions between molecules, and we will end up to networks, so biological networks. And we will study how these biological networks work in functional or dysfunctional states, for example, in the presence of point mutations that are associated with diseases. And we will focus on three main types of networks, which are made of these biological macromolecules. So signal transduction, very important for regulating a variety of processes, metabolism, essential for life as well, and regulation of gene expression. So these are the three main biological processes that we will investigate using this bottom-up strategies, from the very molecular point of view up to the network level. And we will necessarily use an approach that is multidisciplinary in this class. So we will use, of course, the lab part, and Professor Marino will tell us something about this, but we will also use the computer to visualize molecules and see how they interact. And so to predict what the effect, for example, of mutation or a post-translational modification could be at the very molecular level. And then I would like Professor Marino to give some words about the lab part.
Thanks. So basically what we are planning to do for our lab module is to organize the students, divide the students into groups and organize a series of lab experiences in which students can utilize several different biochemical and biophysical techniques to investigate, as Daniela said, the interactions between proteins and several other important biological macromolecules, such as lipids and other proteins, DNA, or small ligands such as ions. This will also allow the students to investigate what are the effects of point mutations, which are due to which are associated with inherited diseases, and evaluate what are the effects in terms of interaction with this macromolecule. So interactions with the DNA, for example, or with other ions. And we also will use this flipped classroom approach, so that at the end of each experience, these groups will give a report to the other groups, which will probably will definitely evaluating also the other groups, so that they are the students are also entitled to let's say grade the other groups' work. And finally, as Danielle anticipated, we will also use some computational techniques to actually visualize what these interactions are. And we will take we will take a look at the molecular level, at atomistic resolution of these structures, and what are the effects of these mutations in the interaction. So this is basically what the lab course will be all about.
Thank you, Valero. Okay, I think we will continue with the next course, which is Omics Science in Precision Medicine. And we have Professor Friso for one module, Professor Malerba for the other. Okay, and also Professor Castagna, but I think Professor Friso will speak for both of them. Yes. You have to switch to your presentation.
Good afternoon. I have the pleasure to be part of this course, and hopefully to have students with whom we will be very happy to share some of the knowledge, and even to deepen research in terms also of deepen more the understanding related to the omics science in precision medicine. Well, the course will be divided into two modules. The first one will be more introductory, and I will leave a word to Professor Malerba, and we'll talk a little bit more about all the methods and techniques and some of the basics on genomics, but you'll talk more about that. Our section, which will be the module on clinical proteomics, metabolomics, and epigenomics, that will be taught by myself. I'm Professor Simonetta Friso, and Professor Domenico Girelli, and Professor Annalisa Castagna. The three of us have a different expertise and background, which I think is quite unique, in the sense that we do have some expertise in basic science, at the same time typically what we call the translational science, in terms that we also deal with the clinical part. So we do think that we have this privilege that we will be very happy to share with you students in this course. Just going into a little bit of more detail in this regard. Let's see if it works. Anyway, it's just a very simple description, in the sense that the course, specifically the module on clinical proteomics, metabolomics, and epigenomics, will open up to a look to the future, in the sense of what it refers to the most advanced trends in technologies, to try to understand in a more dynamic way what it means to talk about the basics in terms of how we can use all the understandings and knowledge that we get from the proteomic science, the metabolomic science, as well as the epigenomics, that it's more my own area of expertise, translated to a clinical application, but we will try to do that either with some classes in trying to make them in an interactive way, of course, but also to dedicate a substantial part of the module to laboratory work, and even to have you learn and have you try yourself different methodologies and try to learn them, to apprehend them, and hopefully even to have some examples on how we do in the practical clinical work use them, or the future for this techniques as well. The other, I think we can go on. Let's see. No, well, that should be okay. What's overall the meaning of the course? Certainly to try to make a strong connection between the human biology and some science in human biology and medicine, and the other, I would say more specifically, what it relates to especially some techniques in that refers to the spectrometry method, or some molecular biology more specific methods, and in the last square, which doesn't work, but anyway, we'll dedicate some time even to design some potential projects also, and maybe to get some ideas, some fresh ideas from you to open to potential novel insights in this area. So the idea is to get the possibility to use, to make into practice, in a sense, what is the interaction used by analytical tools for interpreting the biological systems, pathways, to maybe learn even novel pathways that help us to understand what is the pathology, pathophysiology, and then to potentially use them in diagnostic tools, as well as into prevention and hopefully at the end in therapeutic approaches. I will give the word to Professor Giovanni Malerba.
Okay, thank you. And yeah, I will be very brief. So the magic word are omics. I'm really enthusiastic in having the opportunity to join to this course. The idea is that the course will be very interactive with the students. So it's something new, as already said by the colleagues before. So the students will be very enrolled in the activity of the course. My part is regarding omics on genomics and transcriptomics. So the idea is to study the complexity. So from the complexity to the relevant information. So we will give to the students information on how to prepare samples, what are the samples that we need to study, what are the technology, the most up-to-date technology to be used, employed in order to get the results that want to be pursued. And in the end, especially maybe in the first years, but in the second years as well, how to elaborate, handle the information in this translational way that was already exposed, in order again to find out what are the most relevant information in the sea of this data, genetic and transcriptional data that we can collect, in order to find out the relevant information for the form and the shape and the pathway involved in the mechanism under investigation. So I think that just to summarize, the idea is that we are going to offer the students the most up-to-date technologies together with the most up-to-date designs and the way to analyze the data in order to try to give to try to make sense to the complexity that we are studying. And this complexity can come out by many single simple processes that are working together, or other possibility. There could be a relatively small amount of really complex processes that are working. And the task of this course is to teach the student how to detect what are the mechanisms, many but simpler, or small, a few but complex, or a mix of these, in order to be able to interpret in the correct way the results that we will collect.
Okay, thank you very much, Simonetta and Giovanni. I think we will now proceed with the next presentation, of course, which is bioimaging in translational research. Therefore, I will invite Professor Paolo Fabene to explain a little bit about this interesting course.
Everybody, I'm supposed to introduce 3 different courses, so I don't want to be too late for you. So the first one is on bioimaging in translational research, and he will be actually organizing 3 different modules. And the first one is based on theoretical information by Professor Marzola, and from the physical basis of bioimaging, actually. So she will provide information on the theoretical part of ultrasound imaging, on X-ray imaging and nuclear medicine, as well as MRI, which is, of course, probably one of the most inherent to her experimental practice. And this is based on the theory, okay? Then we will just shift to other 2 modules, which are both with the theoretical part and practical part with the students. So the student will be required to run experiment together, and then to analyze data and discuss data as well. So actually the first of these mixed module are made by Maria Teresa Scupoli and Simona Gambino, and he will be inherent to flow cytometry, and actually it's an imaging as well of the cells, of course. And in the theoretical part, they will, of course, learn the historical view and the backgrounds of the techniques, the possible use and application, and how relevant these data can be in our experiments, and actually in the experiments that the students are supposed to run whenever they will start to work. And actually they will be able to test what they are going to learn just running experiment, designing together and planning flow cytometry experiments, make a data acquisition and discussing data together, and trying to copy this data with some background scientific articles which are available in the literature. The last module is the one that it's run by Andreas Barbati and by myself. And again, we do have a theoretical part on cellular imaging and MRI techniques. And actually, of course, this can be just, very proximal to what Pasquina Marzola is doing. So we will try not to cover too much and to overlap too much on these, but it's something which is going further on the point. So from one side, we learn how it's working. In our hand, we will start to see which are the possible mistakes we are applying to the data. So, and also we will try to combine MRI with co-registration of EEG, for instance, and trying to provide a more biological interpretation of the data. After the theoretical part, we will have the laboratory part, in which again the students are required to run experiment and to discuss all the data they obtain with the literature. So that's a very general overview. I don't think we need to go deeper and deeper. Otherwise, we will, finish the year. Yes. Okay. See you soon, actually.
Thank you very much, Professor Panel. Okay, then the next course is Gene and Cellular Therapies. Professors are not here today, so I will give you just a few information. This gene and cellular therapy is made of 2 modules, one being cell therapy, taught by Professor Carlo Visco, and one being genome editing and gene therapy, taught by Professor Alessandra Ruggero. So in cell therapy, you will learn about stem cell-based techniques and also focus on hematopoietic stem cells and CAR T cells. So all that makes very novel approaches to a variety of therapeutic areas, including regenerative medicine, immunotherapy, and malignant neoplasms. So it will be about how you can use cells to actually heal or to actually treat these diseases. And then the second module will be genome editing and gene therapy. And there will be an introduction to gene therapy with a distinction between non-viral vectors and viral vectors, and how can one perform gene editing, as we mentioned at the beginning, to modify the DNA sequence in a certain way, and then how you can apply this into clinical trials in the hematologic, metabolic, neurodegenerative, neuromuscular, and dystrophy fields, and in cystic fibrosis, and also in anti-tumor gene therapy. So a lot of applications of gene therapy to many important tools, many important areas of clinics. And then there will be a practical part, as you see from this table, where you will learn how to deal with cell cultures and how to prepare retroviral and lentiviral vectors to carry genes, and how to perform this CRISPR-Cas-based editing. So that is, in a few words, what the course is about. And then there is also this biomedical data analysis and biostatistics taught by Professor Giuseppe Verlato, who is not here. So again, I have to give you some words about the topic, but now I have to switch to Italian because he wrote me something in Italian and I'm not able now to immediately translate. So then this will be in Italian, and I'm just reading what he wrote about for presenting, Professor Verlato, for presenting his course. Il corso si incentrerà sulla statistica medica e sul contributo dell'intelligenza artificiale alla medicina. Una signora Novax una volta mi ha detto: un mio parente era vaccinato contro il Covid-19, ma è morto lo stesso. È vero che i vaccinati contro il Covid-19 possono morire, però il rischio dei morti negli infetti si riduce del 90-95%. Prima dell'inizio della campagna vaccinale, la letalità del Covid in Italia è stata del 4,8%. Dopo la fine della prima campagna vaccinale, la letalità è scesa allo 0,3%, quindi da 4,8 a 0,3%. La statistica ci insegna a distinguere la legge generale, la vaccinazione protegge dagli effetti gravi del Covid, dalla variabilità casuale. Anche i vaccinati contro il Covid possono morire, soprattutto se anziani, fragili, immunodepressi o con comorbilità. La statistica ci aiuta anche a distinguere quanto il calo della letalità sia dovuto al vaccino e quanto alla comparsa di varianti più diffusive ma meno aggressive. Il machine learning e più in generale l'intelligenza artificiale sono fondamentali per riconoscere dei pattern, dei fenomeni ripetitivi in quelle situazioni in cui sappiamo poco. Abbiamo tante variabili da studiare, ma campioni con pochi soggetti, come nell'ambito della genomica e della proteomica. In uno studio su 44 pazienti con 2 diverse malattie renali, il machine learning ha mostrato che per una diagnosi differenziale tra le 2 malattie è sufficiente l'analisi delle proteine urinarie, consentendo di individuare 5 proteine chiave tra 1529 proteine urinarie. Quindi statistica medica e intelligenza artificiale sono tecniche indispensabili per dei giovani che si accostino alla ricerca biomedica. So this is in Italian, more or less, the philosophy behind this biomedical data analysis and biostatistics course. So we thank also absent professors for giving us some information. And we continue with the last course of the first year, so research management and experimental design. Again, we invite Professor Paolo Fabene, who will talk for him and for Professor Scupoli. And Professor Bortolotti has another module. It's called Research Management, Regulation, and Bioethics. She had to leave, but she will come back, and so she will give you some information about this important regulation, bioethics module when she's back. So, Paolo.
I want to be paid twice.
It's the same.
Okay. Yeah, 0 plus 0. Yeah.
2 times 0 is 0.
Okay, so the course is on the experimental design, and actually we think it's very, very important that we should approach and we should teach our students not really to use systems or to use new the most newer apparatus or so on, but they should know how to use and how we are not building up technicians, we are building up scientists. So science is an idea, and then we do use the techniques to try to test our hypothesis. And so in this module, we will try, which is again with Maria Teresa Scupoli, Maria Elisa Galasso, and with myself, it's trying to provide information of experimental design, so how to design in a very appropriate way an experiment, how to be able to organize the appropriate control group. And actually, it's not bad to understand that we are trying to design and to plan experiment to try to demonstrate that our hypothesis is fake, because this is what science is. We are not trying to demonstrate that we are right, but we are trying to demonstrate that we are actually wrong, and only whenever we cannot actually demonstrate it, we should assume that maybe we are right. And I think that this is very important for our students, and we will try to push a lot on this, and to understand also which are the hidden variables and the possible pitfalls in experimental approach. In particular, from my side, I will try to demonstrate how many hidden variables are on animal models. For instance, if we are using or testing our animal in a wrong environment, we are introducing a lot of noise. And, if I'm just waking up you in at 2 o'clock by night, and they say, what's about, I don't know, 17 per 24, and you will say, okay, I will say, well, the animal is stupid, and actually it's not stupid, it's sleepy. And so why we should apply our behavioral test to animal on the light phase when they are actually sleeping? So we do have a lot of hidden variables in our experiment. And also there is a lack of information in materials and methods on the journal, which is every year less important, paragraph, but actually it's the most important, because all the consideration that the authors are going to draw about their experiments is based on experimental design they have used. So we will try again to discuss about the gender issue in the animal, for instance, again, the environment, and how to try to avoid false interpretation of our data. And I think that this is very important. I'm actually very, very happy that there is such an opportunity.
Thank you very much, Professor Fabene. We will call you again for another course later, but so don't sit too far away. Then I think we switch to the second year. And as I said, the second year can create a specific path. So there are only two classes that you have to take, this first two in the first semester. But then as you see, you have to choose different classes among groups of 4 in the first semester and of 2 in the second. And this choice will determine your essential path in one of the 3 areas that I mentioned. So neuroscience, precision oncology, or laboratory medicine. So the first one, we will briefly, because we are running quite long, I think, we will briefly mention this first one, bioinformatics for precision medicine. So I think there is also, a module again by Professor Giovanni Malerba. There are 2 modules. I will say a few words about the first one that is called molecular modeling and structural biology. So with this module, you will, maybe I will go closer because I think I'm out of the camera and then I move. The first one, as I said, is molecular modeling and structural biology, and you will use computational methods to do molecular modeling that is essential also for drug discovery issues. So you will use methods from bioinformatics and computational chemistry to see how proteins may interact with specific ligands or with other proteins or macromolecules. And you will be able to actually study and simulate these interactions with the computer, and also use different databases that will help you studying this process computationally. So it's this more structural bioinformatics aimed at possible drug discovery or fields. And then there is this other methods, module, computational and functional genomics that Giovanni will explain.
So thank you again. Briefly, the idea is that here the magic word is bioinformatics. So the course will be oriented in a strong usage of computer applied to our field. My field is genetic and genomics. So students will learn how Professor Fabene already stated how to do things. So as already noted in many papers today, we see what are the results, but how things are done are not very well described. The idea of this course is to teach how to do things in order to get the results using bioinformatics. And this will be done for the course in order to, for example, I will just give you some example, to try to make sense to some genetic variant that are every time detected. So now we know the variants. There is in the population a great variability. We have to try to make sense of this variability and to try to find out what are the variants that are related to the variability of the phenotype, the trait, or whatever. And many variants may play a role or not. And one of the big challenges of today is to try to guess if such variants may play a role or not. And using bioinformatics, but of course, this is just the first step of trying to make meaning to the variants. Using bioinformatics, the course will teach how to filter out the variants that are likely not to play any role in the phenotype variability. Let's say, just an example.
Thank you very much, Giovanni. Okay, and then we continue with the next course, which is cancer biology, metabolism, and therapy. And I think Professor Massimo Donadelli will give us some information about this course.
Daniele, thank you also for your great effort for arriving here today. And just a brief comment is, what a pity not to be a student. What a pity not to participate to this fantastic course in biology for translational medicine. Concerning the specific topic of my course that will be held by me and Professor Ilaria Dando, the title is Cancer Biology, Metabolism and Therapy. We will discuss about the main biological hallmarks of cancer. This is a course that is focused on cancer, of course. And the main part of the course will be the description of the metabolic alterations of cancer cells, because cancer is also a metabolic disease. And it is driven by mutations and key crucial mutation that can stimulate the proliferation of these cells, but also this mutation can drive the alteration of energy metabolism of the cancer cells. And in particular, we will focus about the most important alteration of metabolism about carbohydrates, lipids, amino acids, and nucleotides, of course, all the four main classes of macromolecules. And what can I say? Yes, so we will link the alteration of the biology with the alteration of metabolism, and also in the third part is the alteration of the therapy, also because the alteration of metabolism is linked also to chemoresistance. And so we will focus about the possibility to find new approaches, new therapeutic approaches on the basis of the alteration of metabolism. Nowadays, we are in clinics, there are many traditional therapies that are not involved in the alteration of metabolism, but we believe that these had to be considered for the future. And we will also develop some new concepts about the metabolic heterogeneity of the different type of cancers, because we have to think to not to cancer, normal or cancer, but also to different types and subtypes of cancer. This is also another important point to be considered. And we will also discuss about cancer stem cells, and there are important subpopulation of cancer that will have a specific metabolism and alteration, and we will try to find some new also therapeutic opportunities based on the alteration of these cancer types that will be discussed during the course. This is a course that is not considered as a lab. No, it is only a face-to-face and teaching, and I hope it will be interesting for you. Thank you.
All right, so thank you very much, Professor Donadelli. Now it's the presentation of drug and biological discovery by Professor Ilaria Decimo. Thank you.
Good afternoon. It's a pleasure to be here and to be part of this very novel course. The course that I will teach is drugs and biological discovery. The main aim of this course is to provide students, so to provide you with the knowledge, the basic pharmacological knowledge and the critical thinking that is necessary to develop a new drug, from the biological target identification, and then through all the process that start from the preclinical assessment and arrive until the clinical last phase assessment, and then to market authorization. So during the course, we will learn about all the basic principles of pharmacology, how to administer a drug, how a drug can be metabolized, eliminated, and how the body responds to the drug. And then we will have an overview of all the classical drugs that are already available based on molecules. But also we will focus on advanced therapeutic medicinal products. which includes, for example, cells for regenerative medicine, the use of cells for regenerative medicines. So during the course, we will see novel and classical drugs, and then we will also analyze new drug targets that are still at the preclinical level of development or first phase or clinical level of development. And by doing this, you will be challenged also to try to design a novel preclinical study to identify and validate new drug target. Of course, we will do it together in small groups, and I will give you all the material that is necessary to develop such a scale. Okay, so that's it. Thank you.
Thank you very much. Okay, we will continue with this teaching that you can choose. Laboratory medicine is taught by Professor Marco Benatti, who is not here today. So I will briefly mention what he wanted me to tell you. Maybe I have to do like this again. So, so laboratory medicine is conventionally defined as a branch of medicine encompassing the analysis of blood, urine, and many other body fluids. The leading fields of laboratory medicine thereby encompass many analytical techniques such as clinical chemistry, immunochemistry, hematology, hemostasis, separation techniques, as well as molecular biology. Laboratory medicine has a pivotal role in clinical medicine, from screening to diagnosis, prognosis, monitoring, and guiding and monitoring therapy. It is a constantly evolving branch of medicine, and technology represents the common thread. Significant progress has been achieved in the last decades, which has revolutionized the aims of the laboratory medicine, switching from a specimen-centered to patient-centered vision, paving the way toward precision and personalized medicine. The purpose of this course is to present the last discoveries in laboratory medicine and their application in clinical practice. The main topics of the course are the techniques applied to health and disease, the aging, presented from an innovative and unconventional point of view. So this is the description that Professor Benatti gave us. Then I don't know if Professor Moretti is here. No. Then a few words about this hygiene and prevention medicine. Or it will be a course about essentially many important aspects in public health topics. It will be about public health approach, health inequalities, and also it will cover some key concepts like causality, risk, and multifactorial disorders. And it will also provide you with some knowledge about epidemiology and how epidemiology can be used to study big data in public health. And some topics will be touched, like precision healthcare and public health precision. So what are the difference between these two? And also some other important aspects like the concept of risk stratification, will be covered. So it's about essentially this hygiene and prevention medicine. And then again, I'm giving this on behalf of Professor Moretti because I don't think she is here. And then I think we continue with the next course in neurophysiology and neuromodulation, but we have Professor Marco Cambiaghi who will, so I don't have to present again, who will give us some information about this interesting course.
Thank you, Daniele. And it's a pleasure for me to be part of the crew. And this course on neurophysiology and neuromodulation, it looks like it's going to be, a normal course, so frontal teaching, but it does not want to be a one-man show, but it wants to start like this and then finish as a choir or something like that. And also, its name, neurophysiology and neuromodulation, looks like an old-fashioned style, an old-fashioned style title for a course, but actually it doesn't want to be like that. because if, especially, it's not neurophysiology, the ancient word here, but it's neuromodulation. It's the concept, at least. It's much more older than neurophysiology. And as you can see, the historical perspective is going to be an important part, at least at the beginning, in the very first few hours of the course, which will actually deal with the process of communication between neurons. So to create both circuits and networks up to the development of the behavioral aspect. And to do this, we will go deep into some aspects directly linked to both neurophysiology and neuromodulation. I don't want to go into too much details of these, but of course we will deal with the origins of neurophysiology and the complexity of brain networks. Also, when considering very simple models. Communication between neurons, as I said, but this is follow what Professor Busetto will tell you the first year, and then both neural and glial cells. We will deal with all main extracellular recording and extracellular recording techniques, ranging from LFP to single unit. And with regard to neuromodulation and neurostimulation, we will mainly deal with both invasive and non-invasive neuromodulatory techniques up to optogenetics and chemogenetics. So to understand what it means to be able to control, I don't want to say a single cell, but a very small group of cells, which will induce a change or a modification in a behavioral aspect or in something like that. So it's going to be curious to do it with you, because I do want to do with you. As I told you, it doesn't want only me to talk, but in the end of the course, it's going to be a discussion or something like that in my mind.
Okay,
thank you.
Thank you, Marco. Thank you very much. Okay, and then there is, in the second semester, there is a choice between these two courses. So I will invite, I think, again, Professor Paolo Fabene, maybe for the systems and clinical neurobiology for the third and last time. And he will give us some words about this, systems and clinical neurobiology.
I promise the last one. So, I don't know, I forgot it. Okay, so yes, we are introducing now the neurobiology system and clinical neurobiology. System neurobiology, I will try to share some information related to how great it is to study brain, how 1 + 1 is 3 in the brain, and it's something which is making me excited every time. And it's more like not just like the basic neuroanatomy, in which you start to read the name of the nucleus and then to say, well, this is the pathway and this is the input and the output. Otherwise, everybody will still, sleep immediately. And but it's more trying to understand how it can be possible that different areas are working together and how they are working together in order to, for instance, have a higher cognitive function and so on. So we will talk about the consciousness in the cortex, and we will and thalamocortical system. We will talk about the ascendant pathways and so on. So we will try to figure out a functional neuroanatomy and neurobiology. So we will try to understand how the nervous system is working, thanks to the basis that we again we have seen with the course of neurophysiology. So we will try to figure out how all the information in the neurophysiology can be merged in order with the neuroanatomy, in order to have some, knowledge about how the higher cognitive function are possible. And I'm very happy that the clinical part with Gianluigi Zanusso, which actually is, I think is one incredible person, because he's very great in clinical work and in science and in preclinical also and research. So he will be the perfect person in order to, try to get the first information we got together and try to see how they are working in dementia, for instance, or in all the pathologies that can be related to the nervous system. So we will try again to work a lot in symbiosis in order to be sure that the overlapping material, it's not just a repetition of the information, but just we will try to see with different glasses the same part. Okay. Thank you.
Thank you.
I'm really, I'm not coming back.
Thank you very much, Professor Fabena. Okay, then you can also choose, as I mentioned before, a different topic. If you like to study oncology, for example, this course in precision oncology, which is taught by Professor Andrea Mafficini, who will tell us a few words about the course. Thank you.
Good afternoon, everybody. Thank you for inviting me. And precision oncology, it's a huge topic, of course, and we will try to tackle that with a bit of humility, but also trying to take and rely on all the information that came from the other courses that the student had to be learned from before. So when you deal with this kind of matter, basically you have to collect all the information you can have from the pathologist, from the molecular studies, from all the omics technologies, with only one final aim, which is the decision that the clinical oncologist has to perform to treat the patient. So this is the focus we will use for this course, because as you may know, of course, oncology has been one of the first branches of medicine where the idea of personalized treatment and then later on precision medicine took root. So, and this has been possible because a lot of targeted molecules have been developed in recent years, tested on many patients. And of course, during this process, the oncologists and also the other researchers learned that not all the tumors in all the patient can respond equally to these drugs. So it has become more and more important to subdefine these tumors and subclassify these patients so that the correct drug can actually reach the correct patient. And this has been possible, of course, thanks to the use of high throughput technologies of any kind. So all the omics sciences, and thanks to the evolution of the bioinformatic analysis. So as you can see, there's a bit of everything in this course that has to be tackled. And of course, these kind of technologies can be applied not only on the tumor samples, but they can also be applied on blood, for example. And this enabled also the possibility to follow the patient before the treatment and during and after the treatment to monitor the residual disease. So even this will be one of the topic of the course. The problem is that despite the large amount of data that can be generated from these technologies, there are still many issues with the integration of all this information. And also there's a problem that much of this information can get lost in translation, so to speak. So when you have to finalize this data and get them to the oncologist to achieve a decision, there are many hurdles in this. One of them, for example, is the possibility to find a common language between all the professionals involved. And as some of you already know, this has been somehow somehow tackled with the constitution of molecular tumor boards, where all the different professionalities meet together and try to be of advice to the oncologist to devise the first line, second line, third line therapies, and so on. So this will be another of the topics of the course. Of course, this science is evolving every day. So probably the standards of today will be different from the standards of next year, so to speak. So this course doesn't want to be one where I'm sitting here on the chair and the students are just listening. There will be probably the first lessons just to introduce the general topic, and then all the rest of the course will be performed as a flipped learning, and with a set of specific tasks that will be given to the students. And another thing, I would like to involve some of the residents in oncology to be a sort of jury for the students, so that they can actually interact with the oncologists and have an idea of how their work is impacting and if they are reaching them efficiently. And I think this is all.
Very much, Professor Mattuccini. And there was still the presentation by Professor Bortolotti about this module, research management, regulation, and bioethics, which is part of research management and experimental design. So thank you.
Good afternoon, and thank you also for the patience. The aim of the course, of my course, is to make aware the students that both the preclinical and clinical research have to be carried out following international and national laws, and also regulations and guidelines. And on this basis, the course will be focused on the general, legislative, and bioethic aspects of preclinical research, and also on the general legislative bioethical aspect of clinical trials. In addition, during the course, I will present the aims and the duties of the Ethics Committee for Clinical Trials, which is very important to perform the research, and also of the Ethics Committee of the University of Verona for Human Research. The way in which the course will be given will be through frontal lessons, since the main contents of the course are legislation, laws, regulation, and so we are forced to give frontal lessons.
Okay, thank you very much. Thank you.